del Grupo Latinoamericano de Histiocytosis, y hoy tenemos como tema central el tema de la conexión molecular genética, y vamos a contar con Daria Osipova, que está conectada ya como tema central, así que hoy voy a ser muy escueto en la presentación para realmente dejar tiempo para todo el resto de las presentaciones y los casos clínicos que vamos a... Esta reunión la hacemos en la plataforma Kudo, que es una plataforma que traduce de manera simultánea a un montón de idiomas. Como ven, hay un listado prácticamente de 60 o 70 idiomas, y esto lo tenemos gracias a nuestra colaboración con la Histiocytosis Association en Estados Unidos, y una iniciativa global que la llamamos Global Histio, y donde participamos un montón de asociaciones de pacientes de diferentes regiones, que nos unimos cada tanto para charlar, para compartir conocimiento, para compartir casos, para hablar sobre diferentes temas. Así que todo esto es gracias a esa partnership que tenemos con la Histiocytosis Association, y estamos muy agradecidos de eso. Ok, sigo. Hoy tenemos conectadas, ok, tenemos inscritas a 75 personas, ¿sí? De 23 países diferentes, lo que hoy tenemos inscritos a un montón de países, que realmente nos hace extrema ilusión tener y convertirnos prácticamente en un referente para todo lo que es histiocytosis, no solo empezamos a nivel latinoamericano, pero como ven aquí, hay aquí ya países como en España, gente de Venezuela, por supuesto, hoy que tenemos a Rusia presente, se han inscrito también de Armenia, de Austria, países latinoamericanos, centroamericanos, por supuesto, también tenemos Polonia, Israel y Turquía, y así que estamos encantados de poder realmente, bueno, transmitir y poder conectarnos con tanta gente. Pasamos a la agenda de hoy. Vamos a cambiar un poco el orden de los que está dispuesto, en que directamente vamos a ir a la charla de Daria Osipova, y luego de su presentación, brevemente hará una introducción Jorge Breyer acerca del protocolo LCH3 y unos comentarios que quiere hacer también sobre la última reunión que mantuvimos sobre el LCH4, el protocolo LCH4. Y luego pasaremos a los casos, a la presentación de casos clínicos. Arrancará la doctora Laura Topolansky con un caso de adultos, y luego pasaremos a los casos pediátricos, que serán el caso de la doctora Marcela Córdoba en Chile y del doctor Diego Rosso en la Argentina. Información sobre la próxima reunión. La próxima reunión la haremos el próximo 20 de marzo, el 20 de marzo, en este mismo horario. Siempre mantendremos horario y esta latitud para que parece que está funcionando bien, y lo haremos, antes nos conectábamos los jueves, ahora vamos probablemente a conectarnos los viernes. Así que siempre es el viernes de la tercera semana cada 2 meses. Y el tema central probablemente que vayamos a presentar va a ser sobre el trabajo que está haciendo la doctora Catalina Lava en la Universidad de Buenos Aires sobre la indometacina. Este trabajo es un trabajo que ha ganado el premio al mejor trabajo en la último meeting de la Histiocyte Society en Seattle, al que pudimos acudir. Así que también al haber tanta influencia del tema de la indometacina en la región, nos parece oportuno que Catalina pueda bueno, presentar cómo va con eso. So, Daria, I don't have anything more to say. Now you can share your presentation and start with yours.
Thank you very much for this opportunity to share our study here. I'm a molecular biologist in Dmitry Rogachev National Research Center, and maybe this presentation will be a bit technical for clinical doctors, so just in case, don't hesitate to ask any questions. And let's begin with a quick overview on LCH pathogenesis. We now have come to the consensus that in both high and low risk LCH, the driver mutation is present in a bone marrow resident myeloid progenitor, regardless of the disease extent. And the LCH presentation, clinical presentation, depends on the ratio of the normal cells and the mutated cells. And in case of, for example, single system lesions or multi-system risk organ minus, patients, the mutant allele burden may be below the limit of detection, but the myeloid progenitors, the mutant myeloid progenitors are still there. And the main driver mutation, the somatic mutation in LCH is BRAF V600E, which occurs in almost 50 or 60% of patients. And secondly, most commonly mutated gene is MAP2K1. And also in some rare cases, there are some mutations in NRAS, HRAS, KRAS genes, and some other, but we'll return to the LCH landscape a bit later. So it's hard. It is hard to discuss the molecular diagnostic approaches isolated from the current treatment approaches. So I will quickly overview that as well, even though I'm not a clinician, I'm a molecular biologist. But as we know, the standard therapeutic approach is LCH4, which is vinblastine plus prednisone. It is effective in most cases, but it doesn't prevent the high relapse rate among patients in the risk organ minus group, and it is insufficient for risk organ positive patients. Also, the combination of high-dose cytarabine plus cladribine is effective in risk organ plus patients, but it is associated with high toxicity. And given the fact that in LCH there is a certain mutated pathway, it led to the use of MEK inhibitors. And very recently this year, it has been published by Jean Donadieu and the huge team of co-authors, including our group. It is a publication of long-term effects of MEK inhibitor. Again, as I mentioned, that quite recently, Jean Donadieu and the big team of co-authors recently published an article, rather not published, it was accepted for publishing on long-term inhibitor therapy. And according to this article, and you can see the figures here from it, the inhibitors therapy has demonstrated high efficacy. However, its discontinuation leads to rapid disease relapse. And also, there are a few unresolved issues associated with the long-term inhibitor therapy, like, for example, lineage switching toward CD3 lymphocytes. It happens when a patient is on long-term inhibitor therapy and suddenly the mutation rate in CD3 becomes higher than in myeloid cells. Also, in general, the lack of data on long-term off-target effects on other organs and systems. Also, the clonal and biological evolution of the disease. Like, for example, we had such patient, he was BRAF V600E positive, and he was on vemurafenib, but then he relapsed while on vemurafenib, which is quite rare. So we had to add trametinib, and it worked. So we suspected the secondary mutation there. And also, the most concerning unresolved issue is the development of neurodegeneration. And according to this article by Jean, the 5-year risk of neurodegeneration is 45%. And neurodegeneration is caused by the circulating senescent myeloid cells that break through the blood-brain barrier and cause neurodegeneration. So considering all that, the effective therapy should aim at eradicating the pathological clone to prevent the relapses and to prevent the neurodegeneration. And considering all that, in our center, we used the combined treatment approach some of you are familiar with that. So the main point here is to combine the efficacy of inhibitor treatment with the curative potential of chemo, but in lower doses to avoid toxicity. And after 3 courses of low-dose chemo, we cancel the inhibitors and proceed with the maintenance chemo only. And we have published the results of this protocol quite a while ago, actually, but the relapse-free survival in risk organ negative patients is around 84%, while the relapse-free survival in risk organ positive patient is 77%, and the overall survival is 100%. And now we're working on an update when I have more than 50 patients on a protocol, and I hope we'll be ready till this summer, and maybe I hope we'll present it on the next Histiocyte Society meeting. So let's continue with the methods of BRAF V600E detection. The cheapest, the easiest one is, of course, Sanger sequencing, and its sensitivity is around 10 or 15%, which is certainly not enough to detect somatic mutations. And to increase the sensitivity, you can use the mutant-specific PCR that amplifies only the mutant allele. And its sensitivity is around 3%, but still it could not be enough when dealing with LCH lesions, because LCH lesions are highly infiltrated with normal cells that do not carry the mutation. And the most sensitive method is droplet digital PCR. The high sensitivity is reached due to the fragmentation of each DNA copy into thousands of droplets, where they amplify independently, and then they are detected also separately. So even if you have just one copy of the mutation, it will be detected in its droplet. And in our lab, we use droplet digital PCR system by Bio-Rad. And here is a table from their practical guidelines. And you can see here that the maximum sensitivity of the method depends on the amount of DNA input in your PCR reaction. So the point here is that the more DNA copies you have in your PCR reaction, the less allele burden you can reliably detect using ddPCR. And in case you have like very small number of positive droplets, like 1 or 2, and you're not sure if it's truly positive or it's a false positive result, you can just check according to this table if your allelic burden is higher or lower, the limit of detection that is according to your DNA input here. So over many years in our lab, we had an opportunity to test many different methods on many different types of the biomaterials, as you can see here on this picture. And patient was considered BRAF positive if the mutation was detected in at least one of those biomaterials. And the key point here is that we did the comparison of the mutation detection rates by method. And in case of Sanger sequencing, the mutation rate was the lowest, and it was even lower than expected for BRAF V600E. So obviously, we proceed using ddPCR only as the most sensitive method. And we all know that the circulating cell-free BRAF V600E is a biomarker in children with LCH. So we decided to test this on our patients, on our data as well. And as you can see here, the BRAF V600E is associated with multi-system disease with risk organ involvement, because most of the patients with risk organ involvement were BRAF positive, while most of the patients with single system disease were BRAF negative. And also we compared the initial allelic load, which was also much higher in patients with risk organ positive course of the disease. So the liquid biopsy is a great tool, and but can it be an alternative to the tissue biopsy for molecular analysis? To answer that question, we did the comparison of BRAF V600E detection at initial diagnosis by liquid versus the standard biopsy. And on this table, the red highlighting indicates the discordant results between the methods. And in most of the cases, the BRAF status in cell-free DNA matched that status in tissue. And in additional 4 cases, the liquid biopsy detected a mutation that was not identified in the tissue biopsy. This could be due to the biopsy mistake, or once again, the lesions in LCH are highly infiltrated with normal cells that do not carry the mutation. And only in 1 patient, the mutation was detected in tissue, but not in cell-free DNA. That was a patient with a single system monofocal lesion. So in general, the answer is yes, liquid biopsy has the potential to be an alternative for molecular analysis. However, LCH remains a histopathological diagnosis, and conventional tissue biopsy is still required. And in cases where invasive procedures are challenging, liquid biopsy can be used to determine molecular status, but it cannot replace tissue biopsy for diagnostic confirmation, of course. And also considering the fact that the pathological clone originates from bone marrow progenitors, that they carry the driver mutation, we also tested BRAF status in myeloid progenitors. For that, we used the cell sorter. We isolate them according to their phenotype. As you can see here, CD34 and CD117. And we also did the comparison of BRAF status among patients with risk organ positive or negative. And again, you can see here that BRAF V600E is indeed associated with a more severe course of the disease, while patients with single system forms didn't have the mutation. And also here, 7 patients without affected bone marrow, without clinically affected bone marrow, they still carried the mutation. And this once again indicates the current understanding of LCH pathogenesis, that the source of the pathological clone are the myeloid progenitor cells. After all the experience we had at initial diagnosis, we decided to use these values as an MRD predictor during our combined therapy approach. In our combined therapy, we have the predetermined time points when we measure the cell-free DNA allelic load. And as you can see here, initially in the group of risk organ positive patients, the allelic load was much higher than in group of the patients with risk organ negative. But then only after 28 days of chemo induction, the allelic load dropped significantly. And then during the therapy, it almost reached 0 in some patients, but in some patients, there were still some fluctuations. But the thing is, these fluctuations, they doesn't necessarily lead to a systemic relapse, because they can be maintained by some residual cells and tissues. But here you can see the only patient who exhibited systemic relapse. He was with risk organ involvement. He had the highest allelic load. So in general, the rare incidence of relapses on our combined treatment doesn't allow us to establish any clinically relevant cutoffs, any clinically relevant values. But for sure, the cell-free BRAF load reflects the therapy efficacy and the disease burden. And also considering that the myeloid progenitors are the source of the pathological clone and the therapy should aim to eradicate those cells, we also monitored the allelic load in myeloid progenitor cells. And the same as in cell-free DNA, Here you can see that in risk organ positive patients, the allelic load was higher than in risk organ negative patient before the treatment initiation. But then it dropped and almost reached zero in most of the patients. But the only patient, again, who had the systemic relapse involvement with risk organ involvement, he had the highest allelic load by the end of the therapy. So in general, yes, BRAF V600E load reflects the disease burden and response to therapy, but it cannot yet be used reliably as an MRD predictor. But the main reason behind it is that we have really rare incidents of relapses in general in our protocol. And also, as I mentioned, we're working on an update on 50 patients, so probably we'll have some new information on it. And also, we have many patients who were not receiving the combined treatment, but they were on long-term inhibitor therapy. And in such cases, we also monitored those values in cell-free DNA, in bone marrow myeloid progenitors, in bone marrow histiocytes, and bone marrow CD3 lymphocytes. And at some point, we had to come up with how to cancel the inhibitors. So we decided to cancel them after we see 2 consecutive zeros in both cell-free DNA and myeloid progenitors as the source of the pathological clone. And after that, when we saw 2 consecutive zeros in 4 patients, we canceled the inhibitors, and all those patients, they remain asymptomatic and with no clinical or laboratory signs of relapse. And the median follow-up here is almost 2 years now. And 2 patients here, they still had some allelic load in bone marrow CD3 lymphocytes. But the thing is, as I mentioned earlier, this CD3 lineage switch is well described in LCH and in this article here. But those cells are basically nothing to worry about. Yes, they carry the mutation, but CD3 cells, they do not participate in LCH pathogenesis directly, so they do no harm, even though they carry the mutation. And also the BRAF itself is less expressed in CD3 lymphocytes rather than in myeloid cells. And also those cells, they don't just pop up suddenly. They've always been there. It's just that lymphocytes are much more resistant to the inhibitors than myeloid cells. And that's why the mutation burden grows in CD3 while it drops in all the myeloid cells. So basically, this is nothing to worry about as far as we know. Now we continue to the BRAF V600E negative patients, which is actually almost half of the cohort. In our center, we developed the targeted NGS panel that includes genes that were described in LCH. You can see the list here. And the number of genes is not too big, but the reason behind it is that the less genes you sequence, the higher read depth in NGS you can achieve. And it is highly important when you're trying to detect somatic mutation with a low allelic burden. And to even increase the sensitivity of this method and the reliability of this method, we used the so-called unique molecular identifiers, or UMIs, that are ligated to DNA fragments prior to PCR amplification. And as you can see here on the right, if each read with the same UMI has the mutation, then that is a true variant. But if you can see the mutation in just several reads, 1 or 2, and all the other reads with the same UMI do not have this mutation, then it's a false positive result. This could be just a sequencer mistake. And so far, we have sequenced 93 patients. And a driver variant was identified in 82% of them. And this demonstrates that our panel is really effective, and even though it has quite small number of genes, but they're very focused on LCH. And 7 patients appeared to have BRAF V600E, but those were the cases with low allelic burden that could not be detected by standard methods like mutant-specific PCR and Sanger sequencing. And this once again indicates that we should use the most sensitive method when we detect BRAF V600E. And also 3 patients had another point mutation at the same point as BRAF V600E. They were not initially detected by ddPCR because the probes are V600E specific. And also the very common alteration in BRAF gene are in frame 15 nucleotide deletions. You can see here the screenshot from Genome Viewer, and those deletions are different by 1 or 2 nucleotides. They're still quite similar, but this means that if you want to detect them by ddPCR, you need to use 3 different probes for that. And those deletions are very well described. They activate the pathway and they're associated with the resistance to BRAF inhibitors, which makes sense because they affect the different exon and therefore the different BRAF domain than BRAF V600E. And also we observed a duplication of 9 nucleotides at the splice site of the 12th exon of BRAF that results in the insertion of 3 amino acids. This is how it looks like on Genome Viewer. And this mutation activates the signaling, and it is resistant to the inhibitors. And it was observed in single system bone LCH as well as in 3 of our cases, but still the number of patients is quite small to establish any relevant clinical genomic associations for that. And the second most commonly mutated gene in LCH is MAP2K1. And this gene has 2 main hotspots, which are exon 2 and exon 3. As you can see here in the screenshot, it has quite the same situation as in deletions in BRAF gene. Those deletions are quite similar, but still different by 1 or 2 nucleotides, even though these 3 different deletions result in the same amino acid deletions. But still, if you want to design the probes for ddPCR, you need to do it to do 3 different probes. And also we have patients who are MAP2K1 positive, and we decided to apply the same approach as we do for BRAF V600E positive patients. And we had a case of multi-system refractory BRAF negative LCH who progressed on LCH4. So we applied the combined treatment approach again, but using trametinib instead of vemurafenib. This patient had 15 nucleotide deletion here, and we designed the probes for ddPCR, and we did the monitoring of the allelic load the same as we do in our combined treatment with vemurafenib. And as you can see, the allelic load also dropped very significantly after therapy initiation. And after the first cycle of combined treatment, we observed the rapid reduction of all lesions. And by the end of the protocol, the patient attained a complete remission, and he's still in remission for almost a year now. And also in 18 patients, we didn't find any significant variants. And here on this slide, there is a great example of why we use the UMI barcodes. So here on the left is a visualization of a variant in a hotspot of NRAS gene. And you can see here that the variant allele frequency is around 3%, which is quite enough and quite common in LCH. And also the sequencing depth is enough. It's almost 1,000 reads. So this variant looks fine here, but when you reanalyze the data, but using the UMI barcodes, you can see here that not all of the reads with the same UMI barcode have this variant. And this indicates that this variant is false positive. It is a sequencer mistake. So here is an overall look on our cohort, and here is a big number of patients, 72 patients for whom the NGS was not performed, but the initial analysis showed no BRAF V600E mutation. And for some of the patients, the NGS was not possible because of the low DNA quality. And all of those cases were bone lesions. So the quality of DNA, when you isolate the DNA from bone lesions, is quite often not good enough for NGS. And in other cases, we just had lack of tumor material. And also we calculated the clinical genomic associations, the same as our colleagues do in these publications here. And once again, BRAF V600E is associated with a more severe course of the disease. And also the MAP2K1 alterations are often associated with single system diseases. And more often, they are single system bone lesions with the risk of central nervous system involvement. But overall, there are no significant difference in bones involvement. There might be some difference in lungs involvement. At least BRAF 12 exon deletions are trying to be associated with lungs involvement, but not significantly enough yet. And also BRAF V600E is associated with skin involvement. And last but not least, what if we haven't found any mutations on DNA level? There are still many cases where the driver mutation is fusion, gene fusion that could only be detected using RNA sequencing. So for those patients in our center, we do the targeted RNA sequencing. And we identify in 5 patients the KIF5B ALK fusion, which is very common in LCH and well described in this publication. But in such cases, when you have the ALK fusion, you can start giving the ALK inhibitors. And in this case, you want to start giving inhibitors as soon as possible. So the whole process of DNA sequencing, NGS, then RNA sequencing might take quite a long time. And fluorescent in situ hybridization could be a much faster and cheaper alternative for translocation detections in case of ALK positive histiocytosis. And here are some practical recommendations. So at the initial assessment of BRAF status in patients with LCH, the highly sensitive methods such as droplet digital PCR should be used. Also, the liquid biopsy should be considered as an alternative to standard tissue biopsy for molecular diagnosis, but in cases of localized disease, the results should be additionally verified using conventional biopsy. In the development of new therapeutic protocols, the treatment efficacy should be evaluated by monitoring the mutant allele burden in both cell-free DNA and the bone marrow myeloid progenitor population, which are the source of the pathological clone. And in patients without BRAF V600E mutation, further search for driver somatic mutation should be performed using targeted NGS with high rib depth. And bone tissue is not recommended as a source of biomaterial. And during the data analysis, the potentially low variant allele frequency should be taken into account, and genetic findings should be validated using UMI barcoding or other methods like PCR amplification, for example. And if no mutation is identified by targeted DNA sequencing, the search for driver events should be continued using RNA sequencing or fluorescent in situ hybridization if you skip the sequencing part. And here is just a short overview on those practical recommendations. So once again, you should start with the determination of BRAF status by the most sensitive method. And then in case you have BRAF V600E detection, you should proceed to monitor the allelic burden in cell-free DNA and myeloid progenitors. And in case of BRAF negative patients, you should use the targeted NGS with high sequencing depth. And if no causative variants are identified, then you can do the RNA sequencing or fluorescent in situ hybridization. So that's it. Here is a picture of our center and my lab. Thank you very much for your attention. You can ask any questions, and here are my contact details. So you can contact me anytime as well.